Okay, so we'll start with the intro first. So welcome to the Cambridge Cheminformatics meeting today. Just a few minutes of an intro first, and then we'll come to the presentations. I'm Andreas Bender, so I'm a group leader at Cambridge University, as well as Director for Digital Life Sciences here at Novi Sad in Berlin. And so we started this lecture series about 10 years ago or so. And so the idea was to bring cheminformaticians together. And that means two things, right? So scientifically to talk about interesting science, but also in person, because it was quite apparent that sometimes even people who live in the same town, and Cambridge isn't that large overall, so don't really talk to each other that often. So the idea was to bring people together every 3 months or so, go to the pub afterwards. So that doesn't work right now, but we did that for 8 or 9 years. So it was great to establish that community. So we had physical meetings in Cambridge. And so there was 40, 50 people, and now it's usually about 200 people or so register. And usually it's maybe half or so turn up. So it's usually about 100 or so people attending the meetings, and also from countries all over the world. So usually from Japan to Brazil and many other countries as well. So welcome everyone, especially if you hail from further away. And so to announce also the future dates, so this is the last meeting this year. What we try next year is that we will have 3 meetings on Zoom. So 2nd of February, 1st of June, 2nd of November. It's on Wednesdays again at 4 p.m. UK time. And in between, we also try to have meetings in person again, because I was contacted by a few people who said it would be actually great, if possible, depending on the situation, to meet again in person and to have lectures and go to the pub afterwards. And so those meetings to be confirmed to see if they can really take place, but they would be on the 6th of April and the 7th of September. Yeah, and so there will be people from my group or Chris Swain, for example, volunteer to organize those meetings as well. So thanks a lot for that. You can write down, note those dates already. 3 Zoom meetings, they will certainly take place, and hopefully then also 2 meetings in person. And so you can see all the dates, registration details, and so on this website, c-inf.net. There's also the Cambridge Cheminformatics newsletter, so many of you will be subscribed to that, where I circulate information on new jobs, events, and so on. So that you can see on this website, drugdiscovery.net, there are the previous issues of the newsletter. Or you can also, if you wish, or if you use Twitter, follow me on Twitter, so you will always receive invites, also via the mailing list, whatever your preferred communication channel is. So the main meeting, this one, will generally be recorded, so I would like to let everyone know. And the parts where the speakers agree will also be put online. So there's a Cheminformatics YouTube channel, and that's linked from the c-inf.net website. So where afterwards the recordings can be seen, especially good for people in other time zones. And usually the recordings have a few hundred views afterwards as well. So it's great to make that accessible for others, I think. So the parts where the speakers agree will be made available online afterwards as well in a few days. As for questions, so please type your questions in the chat window if you wish, also along the presentation, and we will go through them orally at the end of the presentation as time allows. And then speakers would ideally follow up by chat for the remaining questions, so that all questions are answered ideally at the end. I will moderate the questions and might ask you to speak and up as well. Use your microphone afterwards if you're asking a question, simply to have some dialogue as well between people who attend today. Yeah, important, of course, put your beer or preferred drink into the fridge, because after this talk, what we'll do is worldwide Cheminformatics pub night. People are attending here. And then afterwards, I will put people into breakout rooms. So 6, 7 people randomly together, yeah. So you can meet old friends again and make new ones, and just have a chat as long as you wish. And in case you are alone in your breakout room because people leave or so, I will see that here. I will put you together with other people again, yeah. So the equivalent of going to a new table, I guess. So stay online afterwards as well. So there will be breakout rooms. You can meet a few people from the area again. Yeah, and if you wish to present, so those meetings are open to all. Please just let me know if you can be work in progress, can be whatever, what your PhD student in the group did and so on. Or if you have information to circulate as well, job ads and so on, please let me know. Very happy to include them in the next newsletter. And there's also one more meeting tomorrow, which I would like to announce. So it's called Digital Sciences for Drug Discovery. So that's a meeting series, a seminar series we organize in Berlin. And you can register for that at this website here, DigiDrug, Digital Drug Discovery, digidrug.net. And so there are 2 talks tomorrow, the revolution of drug discovery. So that's about involving chemists, for example, into using computational tools. Yeah. So how can that be done? That's Nick Stiefel's talk from Novartis. And at GSK, they established the Bradshaw system over the last decade or so. And so Ian Wall will share how they actually establish that system and how they use it within GSK. So feel free to join us tomorrow. That's 4 o'clock Berlin time, 4 o'clock tomorrow, and you can go to digidrug.net to register if you're interested in that as well. But to come back to today's meeting and the actual talks, so our first talk will be on chemical structure generation. So we changed the order of the talks because Aziz has an important appointment exam to take, so he asked to go first. So it will first be Aziz talking on structure generation. Afterwards, Jack Scannell will talk about the predictive validity of models in drug R&D. And finally, Anton Martinsson from AstraZeneca will talk about modeling human in vivo PK directly based on structure and using machine learning. Yeah, and so that is my introduction. So thanks a lot, everyone, for joining today. I would like to hand over to Aziz first of all. So he will talk about on MAYGEN, an open source chemical structure generator. Yeah, Aziz, feel free to share your screen and to guide us through your presentation.
Thank you for the nice introduction, Andreas. And yeah, can you see my screen now?
Yeah, it looks good. Just make it a full screen or presentation mode, and then we should be ready.
Is that okay now? Can you see the full screen? Okay.
Looks very well.
So hello, everyone. Thank you for attending to my talk. Today, I will talk about MAYGEN, an open source chemical structure generator or the chemical graph generator. The outlines of my talk, the introduction, the brief or the summary of the literature about the chemical graph generators. I will just introduce what are these chemical graph generators, why do we need them? And then I will also introduce MAYGEN paper and the good results from our study. But of course, before introducing MAYGEN, I would like to introduce also myself. So I'm from Istanbul. Then I did my bachelor in mathematics, Martinsson Fine Arts University. And then I did my master degree in Boğaziçi University in the computational sciences and engineering, especially Cheminformatics. So as a combination of mathematics and Cheminformatics, that was a great chance for me. And I joined to Steinbeck group. Professor Steinbeck is also here. So I joined the Steinbeck group, Friedrich Schiller University, Jena. So I'm currently a PhD student there, and I'm writing my thesis and my papers. So I'm almost at the end of my PhD, I can say. So how did we start with the molecular structure generators? The whole journey actually started in the 60s. With the NASA project, looking for life on Mars, and then getting all the unknown molecular, getting all the spectral data for the unknown molecules out of our planet. And then for the structure elucidation, they developed this software called the Dendril. It was a part of the first artificial intelligence project in chemistry, the Dendril project. And the aim was we need to get all the spectral data from Mars and then doing the structure elucidation to understand the molecular structures, their structures. But what is the structure elucidation? Just to explain with a simple example, this is not an unknown, but a well-known molecule. This is the NMR spectral data of caffeine. And our purpose in brief, if you have the molecular formula and if you have the spectral data, also, if you're lucky, maybe you will get the substructure information from then from the NMR, the spectral information, we are trying to guess in a combinatorial way, generation of all possible structures and guessing what is the correct or the potential structure, the molecular structure of the unknown molecule. This is what we are talking about, the structure elucidation, or what we mean with the structure elucidation, really a simple, basic explanation. Many chemical graph generators are either breadth first search or depth first search. Here we have a good illustration of the breadth first search algorithms. If we have just the molecular formula, so no bond information and no substructure information, what we have is just a set of atoms. And this disconnected set of atoms, we need to, in a combinatorial way, in all possible way, we need to look for all the interactions and putting the bonds. So it's kind of like playing with Legos or just like a game, but in a combinatorial way, you need to extend and add the bonds in all possible way until the full saturation, as shown here, like a saturated structure 1, 2, 3, etc. But the importance is, or the main problem in the field is the combinatorial explosion. So if we have, for example, C6H6, a small molecular formula, then we have 217 unique structures, the isomers. So it looks like okay, but what about if we have something big or bigger, a natural product called Istanbulin? If you have the Istanbulin with C15H20O4, this natural product, the construction of all possible isomers, it will take days. I know very well because I'm still running on the server. Just I'm curious what I will get with some molecular formula in total having the 19 heavy atoms. So it takes days. And this is a good example of this combinatorial increase, let's say, the size of the number of the isomers that we have by increasing the size of our molecular formula. So in the field, we have the two main methods. One is the orderly generation, and the second one is the canonical augmentation. For the people who already know NOTI, canonical augmentation is from Professor McKay, and NOTI is a well-known and the fastest software for the graph automorphism calculation and graph isomorphism and canonical representation of the graphs. So we can say a graph algorithm tool developed by Professor McKay. So as a good example, but if you are talking about, okay, we have in the field of as the intersection of computer sciences and graph theory, we have the graph algorithms, it's graph generators, orderly generation and canonical augmentation. But in the field of the molecular structure generation, as a good example for orderly generation, we have MAYGEN, and for canonical augmentation, we have OMG. But actually, OMG is not fast, a good, efficient software, although NOTI is the fastest fastest graph algorithm tool. The problem is the lack of good applications of NOTI during the molecular structure generation. So I'm not comparing MAYGEN against OMG, but I'm just giving two examples for orderly generation and NOTI. So just to summarize again what we had or how we started the journey of chemical graph generators, it started with Dendril, and then as open source structure generator, we had open molecule generator and parallel molecule generator, PMG, as a successor of OMG. But in the field, the fastest structure generator is, or we can say until the yesterday night, was MAYGEN, as the fastest efficient molecular structure generator developed by a mathematician from Bayreuth University. The disadvantages, it's commercial and closed source. So my main topic in my PhD was the development of an open source, fast, efficient, at least in the same magnitude with a MAYGEN, a good starting point for cheminformaticians to understand what is this black box MAYGEN is doing, or in general, how, because Dendril was also orderly generation method. So what is this orderly generation method? How does it work with Dendril, MAYGEN, and how we can do something as open source software for the structure elucidation toolkits, or in general for the molecular structure generation. If you are interested in the field of the chemical graph generation, I can recommend you, by the way, the Wikipedia page. It's actually from our paper, Chemical Graph Generators, published in the PLOS Computational Biology. But because of the PLOS Wiki project, where people are writing the topic pages, and PLOS is creating the Wikipedia or reformatting the paper, the review paper, and sharing also in Wikipedia. That's how we have the chemical graph generator on Wikipedia. Either you can read the article, it's actually the same from PLOS Computational Biology, or simply just you can just look at the Wikipedia and search for chemical graph generator as a good review on chemical graph generators. So we need actually an efficient open source structure generator, and from my thesis, the first structure generator was MAYGEN. MAYGEN is, again, based on the orderly generation principle, same as the many other structure generators. So just without going into the mathematical details, nothing, just to mention, the idea is using the lexicographical ordering. Somehow we need to put the ordering to detect when we construct molecules. How do we construct the molecules? Just as the adjacency matrices. If we think a molecule like a graph, And we have the adjacency matrix as the representation. So the idea is the generation of adjacency matrices, and then we need to eliminate the duplicates. So here we deal with the block representation. The first image you see this for the matrix A, A1, A2, AT, all these blocks. Here also we see another image, a good representation of the starting point of MAYGEN. We have this block representation based on our initial atom partitions. But just to make it so simple and easy to understand, I added this image to summarize the whole step. So if we put C6H6, what MAYGEN is doing, it looks for the valences. It says, okay, well, I have the 6 carbons with valence 4, and I have 6 hydrogens. I can deal with the 12 by 12, a big matrix, or I can just say, okay, hydrogens come here. I will distribute you first, and then I will deal just 6 by 6 matrix, and it is better for us. So first it does the hydrogen distribution. So these are the yellow boxes are the all possible hydrogen distributions. But if you already distributed, then that means you already changed the valences. Then we have the new degrees for our carbons. So for example, in the first we have the 4 valence, and then the last 2 are just 1 because there's already the connected 3 hydrogens to each carbon atom. So we have, because of the hydrogen distribution, for example, for the first distribution, that we have the 4-2 for the carbons, the atom partitioning. We can't say these carbons are the same because we already attached some hydrogen. So you see at the bottom, this red boxes, these are just for the atom partitioning for each degree representation here that we have after the hydrogen distribution. And here actually we use this partitioning. It looks so abstract, and that's why I put this atom partitioning here. So for each atom partitioning, we divide actually our matrix into, for example, for the first 4 and 2. But why is that important? I will explain in more details, but in a brief way without the details of the mathematical part. So the idea of MAYGEN has 2 steps. The candidate matrix generation. So we generate the candidate matrix. We don't care if it's a duplicate or not. Based on our valences, starting at the maximum possibility of the bond entry, the bond multiplicity that we can add, we start from the maximum to minimum, filling the matrix in all possible way and generating the candidate matrices. And then when we fill the matrix, we need to perform the canonical tests. And this continues like in a recursive way, the whole process. But for more details, this is just like a simple explanation of what the MAYGEN is doing. And this is more detailed way of like the workflow of MAYGEN algorithm. So at the beginning, we have the molecular formula. We distribute the hydrogens. And then we have the, for each hydrogen distribution, the new degrees and the atom partitions. And that helps us, as shown on the right side, this block representation of matrix after getting the atom partition. Then we start filling all blocks. The importance here is you can just generate all possible matrices, okay, but at the end, just waiting until the end of filling the whole matrix and then performing the canonical test, it won't make the algorithm efficient. So we need an early boundary condition, which is the canonical test based on the blocks. That's why we need the first blocks. We say, we know from the beginning, for example, the first 4 carbons are the same guys and they have the same valences. I need to check whether this block is already in the maximal form or not. If it's not maximum, then just ignore it and continue updating your matrix. Whatever you add after the first block, when you perform at the end the canonical test, you will start from the beginning and you already know the first part is not canonical. So that helps and speeds a lot. I can say without these tricks, it can be 14 hours for C10, H16. I remember from my a year ago test. But with these tricks, it can be an hour, or it can be, like I'm just explaining step by step. With these tricks, it goes from 14 hours to 1 hour, 1 hour to seconds, or less than second. So these early boundary conditions, if we can avoid the duplicates as early as possible during the generation, then we make the algorithm faster. This is actually just the text, but I will just briefly tell you what the canonical test is. We have the 3 steps. We look for two rows and we permute the rows. It's just a calculation of the automorphisms of a graph. So it is a graph automorphism problem. If you calculate the graph automorphism or the automorphism group of a graph, and then you need to start from the first row to the last row. That means from the first node to the last node, you need to look for the permutation, the automorphisms, mapping each node to itself. I'm not getting into mathematical part, but need to mention here, by these permutations, we can have either the same row by permuting two rows, we can have the identical rows, or we can have something almost identical with the help of an additional permutation having the identical rows, or we can have totally a smaller permuted matrix. This text, of course, won't help you to understand what this guy is talking about with the canonical test. And let's look at this simple, very simple example. So in the first matrix, we have the initial matrix. We have the permutation 1, 2. We say if we have 1, 2, and if I'm testing 1, the first row, that means I need to check with the second row and I need to compare two rows. But when we look at the permutation, we need to permute the entries of our second row. They are identical. Then I say it's already the automorphism of just this first two nodes. Then I put it in the automorphism list. Or maybe I would guess something like not identical after permuting. Then I need to look, as shown in the B and C, I need to look for a permutation which makes two rows identical to say, yes, this is the automorphism. So what we had is, in the case of B, 1, 2, and at the end we don't have the identical rows. And we say, is that possible to find a permutation which makes them identical? Is that a question? Are they identical or not? Then we found the permutation, for example, here 3, 5. That means permute the entries, the third and the fifth one, it will help you to have the identical. And at the end, we can say 1, 2, 3, 5. This permutation needs to be added to automorphism list because this is the automorphism list for the first node. And this is how it goes from the first to the end, from top to the end. We need to calculate all the permutations. But every step, we are extending the automorphisms that we have in our list. So it helps. And at the end, we say all the automorphisms that we have in the list are the automorphism of the whole graph, not just one node. But in case you can get also a matrix not maximal. So the maximal is the lexicographical comparison of the two rows based on the atom partitioning. So here, for example, if I will perform with 1, 3, then I have this, the third row I need to compare, and I permuted the entries. But if I will permute with 2, 4, 3, 5, then I end up with this row, and they are not identical. And the compared one, the third one, is already bigger than the first one. We need to keep from the top to bottom in a lexicographical ordering. The first one, the former one, is the bigger one, bigger than the next one. So this matrix is not in the maximal form that we already know, and we can say it's not maximal. That helps us to avoid actually the block and say whatever we continue with this block will be non-canonical. Avoid using it, just ignore this matrix and update the matrix and continue with the smaller matrix like this, the block representation. Just two good benchmarking results for MAYGEN. Again, just to remind, MAYGEN is from my PhD. I can say the last 2 years or 2.5 years, I mainly work on MAYGEN after dealing with understanding the whole concept, the literature and all. So the 2.5 years, the last 2.5 years. But the MAYGEN, the first thesis came in the 1995. So the 20 years old, well-maintained, the fastest open source, fastest commercial structure generator, and then the MAYGEN. But the good thing is MAYGEN is just 3 times slower than MAYGEN, and even it gets closer for some molecular formula. And the PMG, the successor of OMG, the parallel molecular generator, we run it and test it in the sequential mode, not in the parallel mode, none of this. So it is, in average, approximately 47 times slower than MAYGEN. So that made MAYGEN the fastest open source structure generator based on the orderly structure generation approach. And there are gaps because PMG wasn't able to even generate the isomers within a day. The other thing is in PMG, they include the different valences of the nitrogen during the generation process. Even the user didn't specify like we want the all valences. Then that's why, just to be fair, we also put the per structure millisecond, but it is still obvious. Even if you look at the milliseconds for per structure, MAYGEN is the fastest open source structure generator and very close to MAYGEN. Again, 20 years compared to 2 years. But why did we call it MAYGEN? I'm not an egoist, glad to give my initials, but long story short, yes, I'm from Istanbul, Turkish, but I come from a Turkish Arab, ethnically speaking, Turkish Arab Kurdish family. So grown at a house with 3 languages. And I kept hearing from my grandma when I was a kid, in Arabic, do not forget you were just a tiny drop of water at the beginning. Whenever you succeed something, not like I'm done, I'm gorgeous, you were just nothing at the beginning. Why do they say that? Because in the old universities called Medrese and Old Turkish, they have this fountain of life. So you see in the image on the right side, the whole university, it is from the 15th century, Southeastern Turkish city. And this 15th century old, the old university from the 15th century, it has the fountain of life. The first drop of water from the fountain represents the birth, and the whole way here represents our life. And then the pool represents the union going back to the universe, like where we are all coming from, or the belief of these scholars from this university. Why do I explain? Because in the MAYGEN paper, we said, in an old Arabic saying, of course, I couldn't write my grandma's sentence here, May refers to a drop of water. And we hope that MAYGEN will be a good contribution to the field and trigger a surge. Please pay attention to this lovely word, surge, and the development of improved and faster versions, eventually rivaling the best closed source solutions and thereby serving the scientific community. Why surge is important, and why did I say MAYGEN was the fastest structure generator until yesterday night? We released yesterday night the surge before my talk, a chemical graph generator based on the canonical augmentation. This is just a screenshot from the GitHub repository, so it is available, public repository, but we will soon submit the preprint and the paper. That's why I didn't want to add any results like a benchmarking against any other software, but I can say it is faster than MAYGEN in the order of magnitudes, not even a magnitude, but in the order of magnitudes. And it is currently, since it is public and available, it is currently the fastest open source structure generator against the commercial tool, I can say. So the whole story, Dendril, when I read the literature, for me, there was a vicious circle of, in the Dendril, there was a pick, okay, done in the 60s, we had this generator, and then not well maintained. And then we had in the 90s, MAYGEN, but commercial, okay, again, the commercial or like a closed source, or we don't have the code or not maintained. There was this circle, but this circle was already broken, first with the MAYGEN paper, the end of the vicious circle of a closed source or not well maintained chemical graph generators. First damage with MAYGEN, and with search, or the MAYGEN triggered this great search, and search is the fastest open source structure generator in the field. So the whole journey of my PhD, the MAYGEN, and then search, that's why in the search logo, we have this tiny drop which triggered the search, referring to the MAYGEN, the first structure generator from my thesis. And this is a nice, simple, another logo for search. Again, referring to this vicious circle of closed source structure generators. It was this vicious circle damaged with a drop of water, which triggered the great search. And as I said in the old university from this Turkish city, at the end, this is just like let's say like a circle, a tiny drop of water triggered the search, but this is just a contribution to the ocean or the infinite ocean of the open science community. And I just wanted to explain, because this is the explanation of the 4.5 years, this tiny, lovely logo. Thank you so much for listening.
Okay, great. Yeah. Thanks a lot, Aziz, for your presentation. Indeed, applause. And congratulations to finishing a PhD as well. Yeah, that's always a major achievement in life. So well done with that as well. Any questions from anyone? So you have this comparison to you say your tool is only 3 times slower, and that's a very good achievement, right? Because you invested short time and you compare it to efforts that were years and years long, right? Do you have any other ideas what their tricks are? Do you have any hunch what that could be, or do you have nothing in? Can you
regarding MAYGEN, or I mean the tricks of MAYGEN?
Yeah, the tricks of MAYGEN which are hidden. I mean, do you have any hunch what that could be?
No, I could actually add it to my software if I would.
Okay.
Actually, but the hard thing was there is actually no source I can say. Okay, there was the one mathematical chemistry book from the Bayreuth team, but not actually explaining the algorithm, and the papers not explaining anything, the theoretical roots. What I did is going back to the first thesis from the 96 to understand the concept there. Also, they didn't explain the tricks. So by reading that, and then I got the idea, like a first generate candidate matrix by following the ideas of the first thesis, and then performing the canonical test as what I understood so far. But yeah, it is commercial, and they are not willing to actually explain all these tricks since it is commercial. But actually after search, maybe there is no need of understanding the tricks of MAYGEN, but we can change our direction to search. It is open source and the fastest.
That's right. That's right. No, well done. Ages ago, I worked on structure generation as well. It's not a trivial business, I have to say. So well done. Yeah. Any comments? Any questions from anyone?
So I do have a question. I'll turn off my video as well. So thanks for a very nice talk. I was just thinking, and I'm no computer scientist by any means or mathematician, but is it possible to parallelize on GPU and kind of massively speed up that way?
Well, actually, when we published the paper, we didn't have the parallelization option. It was I'm talking about MAYGEN. So we released the 1.4. With that release, we published a paper, but right now we have the 1.8, and we already parallelize from the hydrogen distribution step. That's why I didn't put it, because maybe it will be for another paper, but we already made it parallel. If you remember this hydrogen distribution, for every distribution, it's a good point to parallelize it. From there, we parallelize the algorithm. So rather than starting with C6H6, taking after the hydrogen distribution, shrinking the size and looking at each naive atom partition to start the generation. And for search also, we will have later, not now maybe, but soon we will have also the parallelization of search, I can say, for both.
Okay, thank you.
No, excellent. Thanks for making that available as well. Any other comments from anyone? No, great. Thanks a lot, Aziz, then, for your contribution here. Well done. Okay. So yeah, we will come to the second presenter of today then. And then drug discovery, of course, if you go through that the last decades, right? I mean, first drug discovery was done in animals, small series of compounds 50 years ago, right? James Black and so on, Then there was the high throughput wave, right? In the 80s, CombiChem and all that. And there's a question, should we do more or should we do something better, right? And I think Jack might have an opinion on that. So, Jack.
Well, thank you very much. I'll just share my screen and try and get the right one going. Let me see. Okay. Put this onto view slideshow. No, let me see. Slideshow. Okay, from the beginning. Right. Well, so thank you very, very much for inviting me. I felt actually that, I might have relatively little in common with some of this audience, but actually there are a couple of parallels with the last talk, which I'll point out. But actually, before I do, I'll make some disclosures. So I now run a biotech startup called Unify. I've got my own consulting business, and also I've got an academic affiliation with Edinburgh. But now that's out of the way, I will put up a slide that sort of explains the genesis of my interest in what I'm going to talk about. And although in the very dim and distant past, I was a computational neuroscientist, although I hesitate to use the word computational in this company, I really got interested in R&D productivity when I worked in the commercial world. First of all, working for a consulting firm, did a lot of work in the drug industry, and then working in drug and biotech investment. And doing that, I was struck by a very, very ugly contrast between lots of the scientific inputs that seem to be getting hundreds or thousands or billions of times cheaper and more efficient, and the output productivity of the drug industry that was going in the wrong direction. So again, if you look at the top right panel, that's DNA sequencing efficiency, got something like 10 billion times more efficient over a number of decades. X-ray crystallography, maybe 10 to the 4 times more efficient up to 2010. But the number of drugs in the right panel approved per $1 billion spent by the global drug and biotech industry fell by more than 2 orders of magnitude between 1950 and 2010. It has picked up a bit since, and I may talk about that later. And if I don't, you may want to prompt me with a question. But this struck me as sort of ugly and problematic, right? That we could get so much better at doing the individual things that people who did drug R&D thought were important, and I, as a sort of recovering scientist, thought were important. But on the other hand, it was costing us so much more to discover things. And this led me to some diagnostic work, which I won't go into detail, but I'll mention very, very briefly. And I coined a term with some colleagues, which we wrote about in 2012, which we called the better than the Beatles problem, which is a sort of unusual kind of productivity headwind that the drug industry faces and a lot of other industries don't, right? And the drug industry faces this headwind because it's effectively an intellectual property business. And the graph on the bottom right sort of illustrates what I'm talking about. So first of all, I'm going to apologize for my sort of generational ignorance right here, but people my age like the Beatles, and we call it the Beatles problem because the problem that the drug industry faces is a bit like this. Imagine that every new record that was launched had to be better than the Beatles. Imagine that you could download all of the old Beatles records for free, and then imagine that you never got bored listening to the Beatles. Right now, if those circumstances existed, it would be quite hard to commercialize new music. Now, and that economic conjunction happens in the drug industry. And what we've got here is a graph of the proportion of US prescriptions that are for generic drugs. And that's the orange line going up. And we've got a blue line going down, which is the proportion of US prescriptions which are for branded drugs. And what you can see is that the number of branded prescriptions, or the proportion of branded prescriptions, has gone down, and the proportion of generic prescriptions has gone up. And this is because in the therapy areas where you have successful drugs, those drugs go generic. They become virtually free. Doctors don't get bored of prescribing them. So they effectively substantially undermine economic demand for new drugs in those therapy areas, and also raise the competitive hurdles, right? So then on the right-hand side, there's a number of sort of secondary problems. The fact that we've got this ever-improving back catalogue of virtually free and effective drugs in many therapy areas tends to raise regulatory and commercial barriers. That makes clinical trials much bigger, much more expensive, much more complicated. I'm going to argue that actually it's correlated with the retirement of the best screening and disease models, right? That's been a contributory factor. That's probably going to be one of the most important ones in my view. There have been some technological and managerial missteps because I think people didn't really understand the features of R&D that drive productivity as well as they thought they did. And then also for much of the history of the drug industry, although not recently, it made sense to throw money at problems, and that dragged down productivity over time, right? But it's the problems in black 3 and 4 that I'm going to focus on through most of the rest of the talk and how they may be addressed. And so R&D decisions depend on screening and disease models. And here I've got a couple of real screening and disease models. The leftmost one is some tyrosine kinase data. We have a enzyme inhibition measure on the vertical axis, and we've got a thermal stabilization or binding affinity measure on the horizontal axis. Now, in drug R&D, you might use thermal stabilization as a binding affinity measure. because it's cheaper and easier and higher throughput than doing enzyme inhibition. In the middle panel, we've got some tox data, right? So on the horizontal axis, we've got the toxicity of your drugs in vitro, and that's operationalized as the concentration that kills 50% of your spheroids in this case over the clinical Cmax of the drug, that's the clinical dose. And on the vertical axis, we've got an expert ranking of how toxic those drugs are, right? And what you can see is that drugs that kill a lot of the in vitro cells in relation to their clinical dosing tend to have a low score, which in this case means a toxic drug, right? But again, you see there's a correlation, it's not terribly strong. And then what we try and do is we use data like this, you know, model A, model B, and we line them up and make decisions. And we hope that the correlations between those models and clinical utility on the vertical axis progressively enriches the set of therapeutic opportunities. And the therapeutic opportunities we can think of as compounds, or we can think of them as targets or something else, but these are the therapeutic candidates. By testing the candidates in a series of models and hoping that those models correlate with clinical utility in man, we hope we can make good decisions and invest in the red dots, which are the ones that have high clinical utility, and not in the black dots, which are the ones that have low clinical utility. And a sort of parallel with the previous talk here that strikes me is I came to this rather simple formalism by thinking about drug R&D as a formal search process. And I was interested in the parameters that would affect search efficiency, because this seemed to be a rather peculiar search problem where the efficiency on the individual components of the search task seemed to get much more efficient, but one's ability to perform the overall search task, i.e. find drugs that would work in people at low cost, seemed to be going in the other direction. So here it is a little bit more formally. The term PV here means predictive validity, and you can think of predictive validity of a screening or disease model as the notional correlation that would exist if you had infinite money and no ethics, and you could test a very, very large number of therapeutic candidates in your model. That's on the horizontal axis. And then you could put them all into a human and run a large phase 3 trial and measure their clinical utility in man. And we use models because we have some implicit belief that there is a correlation between these two things. And if we didn't, you know, we might as well toss a coin. And the parameterization here is fairly simple. And if you want it in a bit more detail, there's a source paper at the bottom, Scannell and Bosley 2016. But effectively, one posits a correlation coefficient between the score on the model and clinical utility. You know, compounds are drawn from some probability density function where there is a correlation between these two parameters, which one can vary. And then one effectively searches for compounds that are going to cross some clinical utility threshold, i.e. compounds that would work in people. And one looks at the parameter called positive predictive value, which again, this audience will know, but some I talk to won't, is the true positives, which are in the upper right quadrant, TP, divided by the true positives plus false positives. And one can perform a bunch of sensitivity analyses. One can vary the correlation coefficient between the score on the model and clinical utility in man. One can vary the threshold at which things work in man or not, right? i.e. one can make good candidates rare, or one can make good effective candidates common. And one can also vary how far out on the horizontal axis one searches. And that is effectively a throughput parameter, right? Because if one wants to search for compounds that lie a long way to the right on the horizontal axis that score very well on the model, one has to search through more and more candidates to find such a compound. And that is shown on the bottom graphs. So in the high predictive validity model, by effectively sliding our selection threshold rightwards, our positive predictive value goes up because of the high correlation between the score on the model and clinical utility. However, in the low predictive validity model, as we raise the selection threshold and we raise the throughput, our positive predictive value remains low. And effectively what this is saying is that a bad model is a false positive generator, right? And you can't turn it into a good model simply by testing more stuff in it. And one of the major results is summarized on this slide. And if you look at the left-hand panel, what I've done in the left-hand panel is I've said, okay, for a sort of standard set of search parameters that I think is sort of vaguely relevant to drug discovery, one can vary the predictive validity of our model on the horizontal axis, i.e. is it a good model or bad model, and one can vary throughput, and it's a logarithmic vertical axis, so throughput goes from testing 200 items and picking the best one to testing 10 to the 7 items and picking the best one. And then one can just draw contours that represent the positive predictive value as these two parameters vary. And what you see is that changes in the correlation coefficient between the model and the human outcome of interest, I think a lot of biological scientists would regard as quite small, i.e. moving from a correlation coefficient of 0.4 to 0.5 or 0.5 to 0.6, can have as big an effect on productivity as 1 or 2 orders of magnitude change in the number of candidates tested, right? So what this says is if you're searching for rare positives, and by rare, I mean discovery and preclinical steps. This stops being true once you get into the clinic and positives are quite common. But while you're searching for rare positives, relatively small changes in the predictive validity, the correlation between your model output and the human outcome of interest, can be more important than 10 or 100-fold changes in brute force efficiency. And this brings me back to this idea that one of the big headwinds we've had on drug R&D product over the years is the selective retirement of screening and disease models with high predictive validity. So for example, in 1930, 40 and 50, we had some pretty good models. And what happened with those models is they gave us the drugs that were discovered during the golden age of drug discovery. And because they yielded lots of successful drugs, which are now generic, they've rendered themselves redundant commercially. We don't use them anymore. And we're left with the diseases where the models have proven insufficiently predictive. And ironically, we're still in many cases using those models because they don't give us the right answer and they don't retard themselves. Right? One can also map from positive predictive value to financial value. I won't get into too much detail here, but you can kind of apply these positive predictive value numbers to a sort of standard large pharma pipeline and sort of work out how much phase 1 candidates would be worth. And what you can also show is that it's actually quite valuable even to be able to make relatively bad discriminations between better and worse models. And that's shown in the right-hand panel. So for example, if one wanted to discriminate between, the orange line shows, for example, if one is 60% incorrect distinguishing between candidates that were yielded with models of a 0.8 versus 0.65 predictive validity, right? Well, that's going to be worth $30 million per phase 1 candidate, right? So getting a little bit better at distinguishing the quality of models is in quantitative terms quite important. Now, there's a problem here, of course, which is that we don't really know for sure whether the models are any good until we've put loads of drugs into the clinic and 15 years and hundreds of millions or billions of dollars later found out whether those drugs work or not. Wouldn't it be nice if we could evaluate screening and disease models before we invested all that time and money? I'm going to give you an example here, which again I think comes back to the fact that people sometimes assume the factors that drive R&D productivity, and models are sometimes unevaluated. And the concrete example is antimicrobial drug R&D. And there's quite a lot of good history on this. So one can actually go back and try and get a sense as to what happened. But remarkably, the world's first widely used antibiotic was a drug called sulfanilamide, and it was discovered in the 1930s by a guy called Gerhard Domack, who screened some dyestuffs and dyestuff derivatives. And I can't remember whether they're from the BASF or Bayer chemical company, but it was one of the sort of classic German chemical companies that became a drug company. So he tested a couple of hundred drugs And he found something that became very useful. And until penicillin came along, it was probably the best of the antimicrobials out there. Now, between 1995 and 2005, the global drug industry tested well over 10 to the 7 compounds in aggregate against 60 or so different antibacterial targets. And it found not one compound that was worth putting into clinical development. Right? So this is a really stark example of our technology's got so much better, but we can't discover drugs anymore. Well, there's a number of reasons it got harder, right? So the drug industry by 2000 was interested in broad spectrum agents, not narrow spectrum agents. Broad spectrum agents are harder. But, you know, we knew a lot more in 2000 than we did in 1930. We could test a lot more compounds in 2000 than we did in 1930. So why then was the exercise so unsuccessful, despite the fact that brute force efficiency, or the number of compounds tested, went up by an order of 10 to the 5? Right? Well, probably it's because of the abandonment of high validity models, and in this particular instance, the adoption of low validity models. So Domack screened his compounds, or sulfanilamide was actually found in a screen using live mice that had bacterial sepsis. And between 1995 and 2005, the drug industry went all in for target-based drug discovery, which we now know, and arguably should have been obvious at the time, substantially decorrelated the output of the model, or the things you were measuring, from clinical utility in man. And it did that for at least 3 reasons. First, bacteria have got lots of pumps, permeability barriers, etc. And the contents of industrial high throughput screening collections at the time were enriched for the kinds of compounds that don't get into bacteria, right? So your screening hits were predominantly drugs that would not get into a bacteria if that bacteria was a whole bacteria, right? There were also issues around differences between the genes that were essential in vivo and those that were essential in vitro. And then there were some other problems, I think, around the evolution of resistance to drugs that you found using single target-based drug discovery, right? So here, I think it's quite a striking example of where quality beat quantity, and the industry's learned from this, right? And I think it's moved back towards a more sort of phenotypic approach in antimicrobial screening. But how does one avoid problems like this in future? Well, I'm not going to get into these books in any detail, but there is actually a large and very helpful literature on the problem of sort of quasi-subjective or subjective evaluation, right? Yeah, so we can't wait 15 years and have lots of failed trials, but there's a whole bunch of things you can do to try and judge whether the models one is using may have higher or lower predictive validity. And I would particularly recommend here the book on the left and the book on the right. The Funtovitz and Rabbets book is wonderful if you think about measurement and communicating numbers and uncertainty. And the book on the right is just remarkably practical and sort of very easy to use. And again, I don't at this point want to sort of understate how much work goes on to think about whether models are good or bad, right? There clearly is plenty of great work on screening and disease model validity. You know, some companies have moved towards a kind of validity-centric problem choice. I'd say Vertex is an example of a company like that. Human genetic validation has been a huge help. I see we have Anton from AstraZeneca next. Well, certainly from the outside, it looks like AstraZeneca have done quite a lot to effectively reduce sources of decorrelation between scientific results and human clinical utility. They have something called the 5 Rs program. Some of you may know that biomedical science has been through something called a reproducibility crisis. That's been a failure of people to be able to replicate results published in Nature and Science when they actually try and put a bit of industrial money behind it. And that has also triggered a bunch of remedial action. And I think part of that uptick in drug approval since 2010 is related to improvement in model validity as we've sort of genetically sliced and diced cancer and also moved towards rare diseases, which are arguably more modelable. But I do think, though, the application of sort of better model evaluation is very patchy. And I'm going to propose here that actually there are several frameworks that, given the economics, the sort of non-obvious economics, are worth considering for those who haven't yet considered them and those who are consuming or producing model output. the first thing to do is to sort of formally set out criteria which say the extent to which your model recapitulates human biology. And again, I'll point to a framework that does that on the next slide. Something else that one needs to sort of evaluate and formally assess are tests and endpoints. And in my view, there's surprisingly little literature which tells you how to do 1 and 2, right? Number 3 is statistical and experimental hygiene, right? So are your sample sizes big enough? Are you biased? Is it blinded, et cetera, et cetera? There's actually a very, very large literature there and an awful lot of very practical guidance. And there is one other major gap, which I'll allude to, which may come as a surprise to more sort of mathematically and computationally oriented listeners, but is I think very much true amongst biologists, is I think it's a very important concept called domains of validity, which as a biologist, I had never heard of. But in working on model validity, I became increasingly aware. I was introduced actually to a former cosmologist who was a cheminformatician who introduced me to this idea. But it's a very straightforward idea that models are predictive within certain domains and not within others. So for example, Newtonian mechanics is predictive for objects that are substantially less massive than a black hole, moving substantially slower than the speed of light, and as long as they're a lot bigger than an atom, right? So within those parameters, the model Newtonian mechanics works. Outside of those parameters, Newtonian mechanics doesn't, and you should be using something else, right? Biologists, I think, are very reluctant to opine on the validity of their models. So on to a bit more practicality. There are some good examples of evaluation frameworks in the literature. There's a very nice framework which a collaborator of mine, Guy Ferreira, has published with colleagues, which is called FIMD. That's the standardized framework to assess animal models for efficacy assessment. And also there's some particularly nice examples from toxicology, where arguably the data, the historical data, makes model evaluation easier. But I think some of those toxicology examples, if one reads them, one gets a sense as to the kind of evaluation frameworks that one should be applying to efficacy models. I'm going to conclude really with a couple of anecdotes, right? So at the moment, I'm sort of writing a long, detailed, sort of painfully detailed, in fact, review on how to think about and how to evaluate model validity. And I'm just going to give you a few sort of teasers here before I stop. It kind of seems pretty obvious that one should think about things like biological recapitulation, tests and endpoints, statistical and experimental hygiene, and domains of validity. But actually, if one looks at failure in certain areas of R&D, it seems quite clear that people haven't thought about these things, right? So, or at least not enough people have thought about these things. Again, in antimicrobials, the biological recapitulation was shot by the move from screening against whole bacteria to high throughput screening, because you lost the efflux pumps and arguably you got the genetics wrong, right? Ischemic stroke is a really interesting example. There's been a massive translation failure in ischemic stroke, lots and lots of encouraging animal results, which never made it into, which failed in the clinic. There are a number of problems there, but one very interesting one is related to tests and endpoints. In retrospect, obvious, but in the absence of formal evaluation, it wasn't. Many of the drugs that went into the clinic and failed in human clinical trials in stroke were tested in animals 5 or 15 minutes after the animal was effectively given this ischemic stroke, or the animal equivalent of an ischemic stroke. If a human has an ischemic stroke, it's very difficult to give them the drug in anything under 3 hours, and 5 hours may be more normal. It turns out in some cases, when they went back and did the animal work with a 5-hour delay between the ischemic insult and the drugs that didn't work in people didn't work in animals easier. It didn't work in animals either, right? So there's a kind of clear failure of relating the tests and endpoints from the model to the human condition. And the final example I'll talk about is in cancer drugs, where there are 2 very common characteristics of the models that are used in cancer drug discovery and development. one common feature of the models is that they use very, very fast growing cells or cell lines. And another feature of the models is that the cells or cell lines tend to be genetically homogeneous. And most human cancers, or many of them, are relatively slow growing and they're genetically heterogeneous. And what the cancer models have given us is they've given us drugs that are quite good at curing human cancers that look just like the models. But that tends to be a very rare subset of cancers. It tends to be, for example, childhood leukemias, which are fast growing. It tends to be testicular cancer, which is fast growing. It tends to be CML, which is genetically homogeneous, right? And so what we've done is effectively, we've developed cancer models that are not representative of broader cancer. And we've actually got quite good at discovering drugs that look like the models. But actually, we've been surprisingly unsuccessful at treating the bulk of advanced solid cancers, which don't look like the models, right? The domains of validity of the models haven't applied to the bulk of cancers. I think I'll stop there and see if there are any questions.
Great. Thanks a lot, Jack. Yeah, any questions or comments? Yeah. Okay, and I think we have some in the chat actually. Desiree, would you like to read out a question or elaborate on that a little bit?
Yeah, sorry.
Hi, Desiree, how are you? Nice to see you.
I'm fine, thank you.
So I should say that Desiree is one of my collaborators, so I hope you're not going to ask anything too hostile.
No. No, I'm just wondering, my own research into the specifically the animal model selection to show efficacy, I assessed project applications in the Netherlands for where investigators apply to do experiments in animals at the central authority, and I just looked into, okay, what motivation do people give to select model A over B or whatever? Yeah. Well, actually the question why is not asked, so the information is there but all over the place, so it was quite an exercise to get it, but it was actually very traditional, like we are the experts, we have always been using this, our friends are using this is the most model ever used, or we have prior data, or all that kind of things. And apparently the animal welfare body, the ethical committee and the central authority just accepted that is apparently a good reason. And that's also what you see. And of course, I don't know all the data from industry that is not published, but like the what you see in publications that are open access, actually similar experiments, we use this model dot.
Yeah,
I agree. So do you know how this can change?
So I think it changes by people evaluate. So first of all, I'll agree with you, right? So I think availability bias and cost and historical precedent dominate model choice, right? And for example, I have reviewed grants for a UK biomedical agency where arguably the single most important thing for a reviewer to have known about those grants was, why do you believe that the model you're using will give a result that's translatable to humans, right? That was the single most important thing. That information was entirely absent from the grant application. They told us a whole bunch of irrelevant stuff, you know, but the single most important thing we should have known before giving them money, they didn't tell us. So I think part of my motivation, focusing on the economics, is to make make drug companies realize that you make money by getting people to focus more on the model choice. It's by getting venture capital firms to realize, to say people applying for funding, we're not going to fund you unless you can justify the choice of the screening and disease model, because it will be expensive sometimes to switch. It will cause work, it will cause effort. But I think part of the reason I've attacked this, or I'm attacking it from an economic perspective, is that unless people who are paying for the research realize that the quality of what they're going to get is much higher if they care about serious argumentation around screening and disease model choice, if they don't demand it on the supply side, people are going to carry on doing what they're doing.
Yeah, that's true. Yeah. So I also believe that it's indeed the primary responsibility of investigators who's doing the making the ethical weighing to do this experiment, but indeed it should be supported by all the stakeholders in the line, just by asking the question, show me the data, why. Yeah, that's true. Okay, thank you.
Yeah, there's quite a inertia, right? The status quo is kind of a self-fulfilling prophecy nearly.
And I think in academia particularly, because inevitably you get more citations if you do stuff that's like other people, right? So there is a tendency, and the tools become available. It's easier to use a model that lots of other people have used, even if it's a poor, right? And I mean, there's another reason so another sort of set of bodies that I'm starting to talk to are some of the sort of, some of the biomedical charities, for example, because again, one of the problems I hear talking to some of the grant funding bodies is that, someone at an Alzheimer's charity or an Alzheimer's funding institution, the people giving the money don't even have a common framework with which to articulate their views about the models, right? There isn't even a sort of lingua franca. So, two people might agree a model is good, but they might be looking at completely different criteria, right, in making their judgment. No,
yeah, that's right. One part is science, right? The other one is how do you change decision making? How do you translate that into people's behavior?
Yeah.
Absolutely. Is it Desha, you were raising your hand as well. Feel free to ask your question as well. Just notice that.
So I've got a question on chat. Sorry, can I do the chat question first, unless I'm taking too much time? Please, So there's a question: how do you prove a model is superior? I think here I take a sort of Bayesian view, right? So if a model is a little bit better, it's probably more valuable. And at the moment, we use models because we implicitly believe that they're predictive, but we very, very rarely make the basis of our assumptions explicit, right? So I'm not making sort of counsel of perfection here. What I'm saying is that one should evaluate models against a fairly rigorous set of criteria on the reasonable expectation that the models that reasonably look better on more of the criteria are probably better than the models that don't, right? So it's effectively bringing people's Bayesian judgment of their model validity a little bit closer to reality. It's not proving anything, right? It's not a counsel of perfection. Sorry. Now onto your question.
Yeah, it's basically just a similar question in general, that what would be the best model, or how we can move forward to find the best models, either in biology or in such a complex chemical ways. We work here in EI with organoids, for example, which can be as well a bit more complex than cell lines, but there are billions of further steps which we can take, and I think mostly you have answered my question back.
And for organoids, right, there are some quite good examples in talks that I've come across in the literature. So, for example, the IQMPS, which is an industry consortium, have published what I would consider sort of 90%. They're 90% of the way to doing a checklist for evaluating in vitro models for drug-induced liver injury, and they may have produced others. And they just list a bunch of sensible criteria that a reasonable person would say this is, or rather a reasonable expert would say this is what you want to check if your model is a drug-induced liver injury model. So, for example, do the cells produce the right amount of urea, right? Do the cells produce the right amount of albumin? Do they express the right genes? Do they stay alive long enough that you can test drugs in them reliably, right? And then they also have a bunch of drugs which are positive and negative controls, which they say, okay, if you test these drugs in your model, do you get the right answer? So you can actually work out what its positive, what the true positive rates, false positive rates, ROC curve, et cetera, right? Now, toxicity is easy because there's lots of drugs that have been into man. I think for models for indications where that's not the case, you just have to go on sort of sensible, pragmatic criteria, but just be explicit about what they are. And again, the slide I've got now is a nice concrete example, right? So again, one of my collaborators has done a great job in publishing criteria that I think are relevant to animal models of disease in particular, but it's almost like a kind of meta-analysis you can apply to models where you start with a very clear characterization of the human disease state, and then you itemize that, and then you simply count the extent to which one model or different models tick the boxes that an expert in the field would think need to be ticked. And a useful thing with that framework is often it shows that none of the models may be great, but it also shows the strengths and weaknesses. So in this particular case, right, there's an example of a dog model and a mouse model of Duchenne muscular dystrophy. And in terms of sort of mimicking the epidemiology and the symptomatology, the dog actually looks much better, right? It sort of ticks a lot more of the boxes of looking like the human. In terms of the pharmacology, it does much worse. But actually, that's not because in this case, the dog, you test Duchenne drugs in dog and it produces the wrong results. It's that no one's tested Duchenne drugs in the dog, right? So what this says is that if you're interested in Duchenne, what you should do is you should test a few drugs in the dog to see the extent to which it recapitulates human pharmacology. And then you might find that actually you should be doing everything in a dog and not in a mouse.
Okay. Thanks, Dash and Jack. Any final question for Jack? Okay, great. Thanks a lot, Jack.
No, thank you very much for inviting me.
Thanks for your contribution. Much appreciated. Thanks a lot. Cheers, Jack. Okay, and we will come to our third talk, and that's by Anton Martinsson from AstraZeneca. And actually, there's a connection between the previous talk, Jack's talk, as well as Anton's, because the question is always, should you run in vitro models extrapolate to the in vivo situation, right? Or do you model in vivo data directly? And that's in particular for the PK area, what Anton will talk about.
Yeah, so thank you, Andreas. And let's see, so I share the right screen. Slideshow. That was not the right screen. Yeah, are we all good?
Yeah, it looks good. Thanks.
Yeah, great. Yeah, so I'm happy I got the opportunity to present. Also that I presented after Jack, because I think this will tie back quite a bit to it. So my talk will be on machine learning models for predicting human in vivo PK parameters using chemical structure and dose. So before I move into the details, I would like to put this into context. So our group works with producing machine learning models to guide early drug discovery. And traditionally, we have mainly been producing models to predict in vitro assay endpoints. But recently, we've made a shift into trying to predict in vivo endpoints, which of course, the closer you get to human, the better it is, because we want to design drugs for human and not for a cell in an in vitro context. So what we are trying to do here is to do it from only structure and dose. And if we can do that, that can help us to actually do it already at the point of design. So this would help us to drive prioritization of compounds for in vivo assay testing. So that would help us from a 3Rs perspective, not only with making better compounds, which is actually the next point. So that would help us to design compounds with better safety and PK properties. And by doing that, it would improve the speed and efficiency of the design, make, test, analysis cycle here presented on the left. So this is what we're trying to do here. So this talk will be divided roughly into 3 sections. So background and data creation first, followed by modeling and the results, and how we want to, and lastly, how we want to apply these models and some conclusions. So why are we interested in PK? So PK is important to achieve 2 of the 5 Rs that Jack mentioned. So right tissue, which means that the drug needs to reach the right tissue to engage with the target, but also the right safety. So PK can be described as ADME, or absorption, distribution, metabolism, and elimination. And various parameters can be derived here from these kinds of curves on the right. So time versus concentration curves. So for absorption, we are interested in bioavailability, which measures the uptake of drugs orally. For distribution, we're interested in volume of distribution, which measures how much actually reaches the right tissue. And for metabolism and elimination, we're looking mainly at clearance. So for bioavailability, we want to have a high bioavailability to increase the exposure, and for clearance, we typically want to have a low clearance. And in addition to these endpoints, we're also interested in Cmax, so the maximum concentration here in this graph here. So let me just change my pointer. And we want this concentration here to be above the MEC and below the MTC, which makes the therapeutic range so we can achieve both efficacious and safe drugs. So in order to model, of course, we need data, and we extract the data from Elsevier's Pharmapendium, and more specifically the PK module. So the PK module at the time of when we accessed it consisted of over 1.2 million data records of aggregated human clinical data from the FDA and EMA. So this was aggregated and not down to patient resolution, but there was information about things like sex, age, concomitants, and a lot of metadata about the groups. So we created a team of data scientists, but also scientists from DMPK and clinical pharmacology to set up an expert-driven data cleaning protocol. So we had a lot of inclusion and exclusion criteria. I won't be going through them all, but in essence, we restricted this data to healthy adults in volunteers, but also in patients for oncology. We restricted ourselves to single dose PK for compounds with a molecular weight below 750, and also to non-tissue specific measurements. So that's what was included. For inclusion, we dropped data entries with missing data, or if the patient group showed sign of decreased PK functionality, and this could be things like renal impairment, and a whole other list of things. So we reduced this big data set of 1,373 drugs down to 1,001 unique structures. So after we extracted the data, we wanted to also extract our PK parameters and aggregate the data. So we managed to get 9 PK parameters, 12 if you split them into per oral and intravenous, namely the ones presented on the screen here. So for PO, Cmax, Tmax, bioavailability, and estimated volume of distribution. For IV, total clearance and volume of distribution. And for both of them, AUC, which is area under the time concentration curve, half-life, and renal clearance. Since we thought that dose is going to be important when predicting Cmax and AUC, since these are dose driven, we chose to not aggregate down to only one row per compound, but we actually took both dose and compound into account, meaning that in our final data set, a compound can be present multiple times, but at different doses. So when we did this aggregation, we took the median value. The doses were rounded to 2 decimals if the dose was below 10 megs, or to the closest integer if it was above 10 megs. So after doing this, we obtain a final data set of 4,491 dose compound combinations for 12 PK parameters, consisting of 1,001 unique structures. But that's not really the full story, because not all endpoints have equally an equal amount of data, which is presented here in the graph to the right. So I think we can see right away that these higher bars of around 3,000 data points are all oral administered endpoints, and that's not so very surprising because oral drugs are more common than intravenous drugs. And the level of data completeness ranges from 3.5% for renal clearance up to 67% for Cmax PO. I think it's also worth mentioning here that this data here is biased towards compounds with good PK profiles, more than your typical early drug discovery data set, because this is clinical data, meaning that there's a bias towards high bioavailability and low clearance, for instance. In terms of doses, we can observe a wide range of the actual dose, but also the number of doses per compound. So we see the dose here on the left ranging from 0.01 to about 10,000 megs. So it's quite a massive range. The mean dose was 46 megs and the median was 58, which seems reasonable. In terms of number of doses, 1 is the most commonly reported with an exponential decay, and 3 is the median number of doses per compound. So after cleaning the, extracting data, cleaning the data, and then also applying some standardization on the smiles, we generated 4 different feature designs that we benchmarked when doing the model building. The first one is just called dose, and that's because it consists of a single descriptor, which is the dose. And we chose to include this not because we thought this was going to be the most predictive feature set, but rather we wanted to compare this and how it relates to these more complicated feature sets. The second one we're calling 2D descriptors, which is comprised of about 100 1D and 2D descriptors using an internal software called OESELMA at AstraZeneca. The third one is quite an interesting one. So this consists of machine learning in silico predicted values of common ADME assays, as well as predictions of in vivo rat PK. So we're actually using machine learning as an input to this machine learning model. And then finally, we concatenated these 3 sets into a 4th set that we termed combo FP, which is a combination of the others. So after doing a pre-study where we compared random forest and XGBoost, we chose random forest because we didn't see any practical difference between the 2 techniques. So we chose random forest for simplicity. So we did a 5-fold cross-validation and hyperparameter optimization with random forest for all of these feature sets. And when we did this, we applied a custom data splitting strategy for both creating these validation sets, but also to create a 20% whole lot test set. So it was important that we had no overlapping compounds between the sets because, the compounds are non-unique. So we needed to take care of that. So this is the result from the cross-validation for all these different descriptor sets. So each color here on the bar represents one of these descriptor sets here. So I think first off, what we can see is that the combo FP tend to be better than the rest. And it's also very important to see that it is better than those itself. And in some cases, it's not. So here we're not trying to say that we can model everything perfectly. This is kind of honest results of what we could achieve. But we see here on AUC IV that we don't do much better of adding all of these descriptors than just using those. So for that reason, maybe this isn't very appropriate to model. You can see the same here in terms of RMSC. But for instance, for Cmax-PO, we see that we do see an increase, but also for Cmax-PO. The other thing here to note regarding the dose is that all of the non-dose dependent endpoints like clearance or any of the others doesn't really, it isn't predictive at all using those. So that's nice to see. So from this, we concluded that the combo FP was the best performing descriptor set. In terms of the whole lot test set results, we saw the same trend when looking at these different descriptor sets. So here I'm just condensing this down to only combo FP, which is the navy bar here in the middle. But what we also want to do is to put these results into context. So you see these additional dashed bars here. So the mulberry bar is what we call a mean predictor, which would always provide the mean value from the training data set. So this serves as kind of the worst case scenario in terms of performance, and we definitely needed to be better than this. And on the other end of the spectrum, we have the experimental error, which we estimate from standard deviations when we aggregate the data. And we consider this to be kind of the maximum achievable results we can achieve, what the data allows us to do. So there are a few observations that can be made here. So first is actually you could do this analysis before even doing any modeling. And an example of this is the Tmax over here. So even though the RMSE seems to be really low and it appears to be a good model, the difference between the mean predictor and experimental error is very slim. So that kind of raises the question, is it even worth modeling it? And the other thing to note here is also you can have two endpoints with practically the same RMSE. So here AUC PO and clearance IV. And if we were just look at the RMSE, they would look pretty much the same. But when doing this comparison, we see that AUC PO is much closer to the experimental error in comparison to the mean predictor than if we compare it to clearance, which is closer to the mean predictor than experimental error. So this tells us that this has probably more practical utility. So after doing this analysis and looking at scatter plots and looking at a few other metrics, we concluded that AUC PO, Cmax PO, and volume of distribution for IV had satisfactory results, and we wanted to implement these internally and continue the validation. So if we stay on this just quickly, this is the correlation viewpoint of these models. Basically we see that these center around the unit line, even though they are far from perfect predictions, at least they center around identity, which is good to see. In terms of feature importances, I already alluded to it before, but our hypothesis was that for Cmax and AUC, the dose is probably very important. And it turns out that it is. So what we're looking at here is forced feature importances. So if we take a look at these first, we see that dose accounts for approximately 50%, which appears to be a lot. But it also tells us that only using dose only gives you half the story. So applying a machine learning technique has probably some benefits. The other thing we see here is that predicted in vitro endpoints and rat PK in vivo endpoints also appears to be important. So these are the ones beginning with pred for predicted. So that has clear benefits, and we could also see that from when we did the combo FP. So last but not least, we also wanted to benchmark this on internal AstraZeneca data. And this also ties back to like the discussion before about the domain of applicability or validity. So we got a data set from colleagues here at AstraZeneca, Davis et al. That published a paper on the DMPK strategy for the past 20 years. And in this paper, they published 126 compounds with clinical readouts, dose, and also their predicted PK parameters prior to clinic. So this was a very good opportunity for us to benchmark our models, like an additional test to the holdout test set, see how we actually do on AstraZeneca chemistry. And it's also important to mention here that these compounds here was not part of the Pharmapendium training set. So there is no overlap or bias in that way. And these results are presented here on the right. If we start with the peroral endpoints, we see that we do worse in terms of RMSE than the DMPK state-of-the-art method, not using machine learning. For volume of distribution, we see an incredible result, but this is only for 5 compounds. So it's more anecdotal rather than a statistical validation, but nevertheless, it's nice to see that it performed well for these 5, but yeah, we can't say much more about it. But the takeaway here is not that we did worse than DMPK, but it's actually that we do all right, because the RMSE is the same as for the holdout test set, more or less. So it can tell us that the models are robust of also predicting this chemistry. So from this, we can say that the models are fit for purpose to be used in early drug discovery, and the DMPK methods are better, basically prior to going into clinic. So the machine learning and state of the art are complementary to each other. With the unique selling point, of course, that we can actually predict it already at the point of design, which is not possible for DMPK. So how do we want to apply these models? So I talked about it initially, but this can help us increase the time and the efficiency, speed and efficiency of the design, make, test, analysis cycle. So this is now implemented internally at AstraZeneca, and it can be used in large-scale virtual screening applications, as well as for individual project compounds. So it's nice to see that it's actually getting used. So the other part of the story is the safety-related part. And that's actually that we want to move into more sophisticated safety models as well. And to do that, we need some kind of measure of exposure. So while you can use dose, that's not the full story, because as we know, not everything will reach the tissue. So if we can use volume, Cmax, and AUC, maybe we can move towards organ toxicity models, which we hope to do. So to conclude, we established a comprehensive protocol for cleaning human PK data, which can be used for machine learning purposes. And we can do it also from just chemical structure and dose. Among the 12 parameters we extracted, we could train 3 fit-for-purpose models, namely for ACPO, Cmax, and volume of distribution. And we could also successfully validate it on internal AstraZeneca data. While physiological-based models show better performance, their development and validation takes a lot of time. You can do it at later stages compared to the machine learning models. So this gives us a complementary approach to this much earlier. And lastly, our paper on this work was published in Molecular Pharmaceutics 2 weeks ago. So if you want to have a closer look, feel free to read the paper. And with that, I want to thank my colleagues at AstraZeneca, and again, Andreas, for the opportunity to present. And with that, I will hand back to you.
No, thanks, Anton. And I should say that's really the first model that I'm aware of as well, right? That models in vivo human data directly, right? So this is really novel in that field, yeah. Okay, good. There are some questions, or at least, yeah, Gregory, you have the first question. Would you like to elaborate on that? Gregory, do you have a microphone? Or otherwise, I can read out the question as well. Oh, yeah. Okay. So Gregory is asking, did you try to predict dose normalized AUC instead of AUC?
So I alluded to it, or I mentioned it, but we do have an in vivo rat PK model, and in that model, we actually predict Cmax and AUC scaled by dose, or normalized by dose. But here, since we want to see what happens when we use dose as a feature, we chose not to scale it. So that's the reason. And if I'm not mistaken, I think we also tried to compare, like predicting AUC scaled by dose and not including dose as a feature, but I think this was more successful.
Yeah, you can do both, of course, right? Conceptually. Yeah. Mm-hmm. Yeah, that is right. Any other comments or questions?
Can I ask a question, Andreas?
Sure, please, Tara.
Hi. So my question is regarding the way you have used those, because it's a complicated, it is a complicated parameter. So you can, for example, have a dose per day, or you can have one-time dose, or per kilogram body weight, and you know, how did you deal with that? Because I have tried to use those for other purposes.
Yeah,
It's quite complicated.
yeah. So that's a very good question, and that actually took a lot of time to take into account.
Yeah.
So first of all, so not all doses. So we used milligrams in the end, but not all doses were originally in milligrams. But whenever it was possible to convert using, for instance, 70 kilograms as the physiological body weight, we did that. But we also restricted ourselves to only single dose PK here. So that means that it's only one administration. That then we didn't have to worry about multiple doses. Downside of this is, of course, that you lose a lot of data because, it's not
Yes,
very
exactly, because sometimes doses given in per day, divided doses perhaps.
Mhm.
Yeah.
Yeah.
And my other question, if I have time, this is quick. The MPK parameter that you mentioned in one of your slides, is it animal extrapolation?
Yeah.
What,
So
what sort of data is it?
Yeah, so I'm no DMPK scientist, so I don't know exact details, but it's in vitro in vivo extrapolation. And so you have, so that's also pretty much the reason, sorry, it seems to have froze. I had this table on which, okay, now it's delayed, but it's getting here. Yeah, so this one. So these are the in vitro features we used as features here, but these are also the in vitro data that the DMPK used to do this extrapolation. And also with animal data, that prediction gets better. So that was kind of the idea, to mimic the DMPK way of doing it with including these in vitro data predictions.
Right, so it is some in-house method of using a combination of these in vitro and perhaps in vivo data. Is that correct?
Yeah, yeah. So it's more like a mathematical framework rather than a machine learning model. But yeah, so I think it's published. We refer to it in the paper. But
Yes. Thank you. Great work. Thank you very much.
Thank you.
Yeah, and by the way, Vignesh just posted a link to the paper as well, so that it's just been published. So that's in the chat in case you're interested. And Prachi, you had a question regarding dosing as well. It might have been similar to what Tara just asked. Does that answer your question as well, or would you like to elaborate on that?
Yes, you did touch upon that. I guess I was trying to ask if you tried to clean your data, or include data sets where you set a threshold on including data points where you had n number of doses, and if any multiple dosage data points seem to improve the model versus if you chose to exclude the ones with lower dosing data. Does that make sense?
Yeah, it does make sense.
Okay.
So so we didn't exclude if it only had like one dose and one measurement. So it's kind of this graph again. So most compounds only had one dose. And since this data is already aggregated from, to start with, so this isn't one patient. It could be different sizes of the patient group. So I think ranging from, I don't know the lower limit, but up to like 1,000 patients or something. So it's really hard to tell, and sometimes you would have standard deviation within this patient group reported, but most of the time not. So it is difficult, but no. So we didn't restrict on that. And regarding high and low doses, this was also a consideration. So it's a balance between utility and performance, because we want to be able to predict high doses. So we want to include high doses, but at the same time, so we did not include this really high dose here. I think we put the threshold to 1 gram, if I'm not mistaken. So ideally you want to include as much as possible, because that makes your domain of applicability larger.
Okay. Yeah. Thank you.
Yeah, that's right. But to ask the decisions, which data to use, right, and how to use it, that was quite a painful process. I mean, I still remember that as well, right? because of the abstracting process. I mean, sometimes you have data on cohort sizes, sometimes not. Yeah, sometimes you have standard deviation, sometimes not. Right? How do you weigh one study with 1,000 participants? Yeah, and 20 other studies with 5 to 12 each. Yeah. So what do you do in those situations? Right? That was quite a difficult process, right, Anton?
Yeah, it was really, and so we tried different approaches. Kind of, so when you do aggregation, you could imagine that you could weigh the value. So if you have the same compound at the same dose, and you have one study with 1,000 and one with 10, maybe the one with 1,000 patients should weigh 100 times more. So we did try that, but we didn't see any practical.
Yeah, I mean,
doing that.
sorry, yeah, I think that's kind of one of the reasons I asked, because when you start having more data points per dose, you start incorporating more variability. So how does that account into the model, and how is the uncertainty transmitted in your results? In general, I've noticed that when you're finally calculating just, Cmax, the errors seem to diminish eventually. So that's kind of what I was leaning towards in terms of uncertainty in the model. But yeah, no, great work. Thanks.
Yeah, so we did do a similar analysis, like on high versus low doses. I didn't include it here, unfortunately, but it was basically, so you have this plot here on Cmax. Again, my cursor is frozen.
Yeah.
But never mind. So we would we would set a threshold about, I think we put 10 milligrams or 100 milligrams, or basically you could put any threshold, and we would see if high dose Cmax is always predicted as high Cmax, because that's not necessarily true, because you can have poor bioavailability and so on. But we did see that no matter if it was above this high Cmax or high dose threshold, we could actually predict them pretty much equally well. We could also see that you could have a high dose compound with a low Cmax and predicted well as well. So that was great to see.
Yep. All right. Thank you.
Thank you.
That's right. And the last question from Koichi, would you like to elaborate on that?
Oh yeah, thank you. Thank you for your presentation. I'd like to know. I'm not sure, but I remember it seems that our MSC of AUC-PO was lower than that of AUC-IB.
Yeah.
I think the AUC-PO is more complex than AUC-IB. So a little bit strange for me. So if you have, are there any speculation?
Yeah, and I think it's due to this reason. So you have 5 times more data for PO than for IV, which makes it harder to predict. And apart from that, I couldn't tell, because we didn't look very much more close in it. But I have observed previously that actually things like intravenous Cmax, it's a little bit different story, and it's a little bit of a weird endpoint, but that's also harder to predict than Cmax PO.
Okay. Maybe data point.
Yeah, so here the number of data points.
Okay.
Yeah.
I understand. Thank you.
Yeah, it's many more data points. And that means, of course, all performance estimates, are not really comparable either, because you sample the whole space differently. It's not that more data points are better. That also leads to over optimistic estimates of performance, because you always find something close in your data set. But what does that mean for new data, right? So that is another question. It's very difficult to compare that then in the end. It could be due to chemical space of IV compounds, as what Tara said.
Okay.
Indeed. Yes.
I understand. Thank you.
So I think so just to add to that, so I think in addition, so we have this complexity that we don't have much data to validate on, but it's also the fact that these compounds are well characterized good PK compounds. And when we predict early at the point of design, that's a different chemical space, and we can't really validate it, because it takes a lot of years for a fraction of those to finally have an experimental data point. So that's also a very challenging part of this.
Yeah, that's right. Okay, Great. I think we probably should conclude here. Great. Thanks a lot, Anton, for your contribution as
Sorry
well.
to interrupt you, Andreas. I think
Yeah,
you've
more.
missed my question in the chat. If
Okay.
you have time for that, maybe just very quickly.
Yeah,
Anton, I think you at the end
go ahead.
briefly mentioned that you want to, at AstraZeneca, move towards organ-specific models. So does mean that you will try to predict which part of the dose will reach which tissue?
So I don't know if we have established that like specifically, but the idea is to go from, to quote Andreas a little bit, model of models and proxy measures to actual organs, which in the end we want to optimize drugs for. So that's the general idea, and to do that properly, we need to put that into like to a bigger context of PK2. So it's more a direction than like concrete plans right now, I think. So there are some efforts, but yeah.
Okay, thank you. I mean, for what I was suggesting to predict the dose that reaches a specific target, you of course would need data for that, which would need to be generated in the first step. So I could imagine this would be a longer process to get to this point. But yeah, thank you for answering my question.
Yeah, that's right. You would like to have tissue-specific data, right? But that's very scarce. I don't see that coming in the very near future, at least. Oh, that's good. Okay, great. Thanks a lot, Morris, as well. Yeah, thanks Anton for your contribution, and indeed for the contributions of all speakers. Thanks to all the participants as well who are here. Please stay here. So join us again next time, 2nd of February. So now we will go into the breakout rooms, so you can talk to the speakers or meet each other again. So thanks a lot, everyone, for participating, and hope to see you next time. Okay, thanks a lot, but please stay online as well. You will talk to very interesting people in a few seconds. Thanks a lot.
Thank you. Bye.
Thanks.